Hello, I'm Celestin, a final year medical student. I'm very grateful to MAS for the research to publication program brought by BMJ in collaboration with UCSF. I'll be presenting on the topic results, reporting all findings succinctly. So we recall the acronym IMRAD for the different sections of a manuscript, which stands for Introduction, Methods, Results, and Discussion. So what is the results section about? The results section is about answering the research question. It's about presenting what we found in the most simple and clear way possible, even to a non-scientific audience. It's about highlighting only the main findings in the text and reporting fully our study results, whether associations were found or not. So what is the results section not about? It is not about restating all that is already mentioned in figures and tables, because by doing that, we'll just be crowding the manuscript. There's a lot of data found in the figures and the tables, so in the text, we only want to highlight what is most important. Results section is not about presenting only statistically significant findings, because we need to report everything, whether associations were found or not. And finally, it is not about discussing our findings, because there's a section dedicated to that, which is the discussion section. We'll now look at some key tips for good reporting. We need to report results fully and honestly, as pre-specified. We need to use text, tables, and figures appropriately. The textual portion typically follows a logical sequence because it tells a story, and the tables and figures act as evidence. We need to report results for primary outcomes first, and we need to give associated confidence intervals for those main results. We need to report only essential summary statistics such as the mean, the median, and their measures of dispersion. Non-essential summary statistics such as skewness and kurtosis, for example, should be left out. You need not to mention tables and figures which are really not essential. So non-essential tables and figures containing a lot of data should not be mentioned. We should keep only the essential tables and figures, and we should not discuss our findings yet. We need to ensure that we leave the discussion to the discussion section. So it's one more thing that editors, reviewers, and readers may look first and sometimes even only at the titles, the abstracts, tables, and figures. So it's very important that tables and figures should be self-explanatory. They should stand alone and tell a complete story. A reader, just by looking at the tables and figures, should be capable of figuring out what the research is about, the same way he would do so if he was reading the abstract only. We need to keep non-standard abbreviations in footnotes. So these are abbreviations which are specific to a particular field but not known by everyone. Symbols can be used, and we always need to check the author guidelines or journal requirements. Tables capture information concisely and display it efficiently. They should provide information at any desired level of detail and precision. For example, a certain number of decimal places, confidence interval, percentages, and all of that. Everything needs to be mentioned at a desired level of detail and precision, yet in a concise way. And they should have a short yet self-explanatory title. Figures, like tables, should be self-explanatory. They should have a clear and consistent set of annotations. They should have legends, and we have examples of figures include primary evidence such as X-ray films, scans, pictures of pathology, specimens, electron photomicrographs, which are really cool. We have graphs, which include line graphs, bar graphs, scatter plots, box plots, and illustrations such as experimental setups, workflows of participants, hypothetical models. All these should be self-explanatory. They should be clear, and they should have clear and consistent annotations, and they should have legends. We will now look at some common pitfalls in results reporting. And the first one is that scientists tend to write more to obscure rather than to inform. And this is typically what happens when they report in a very extremely technical way, such that it is not really intelligible to the average reader or to a non-scientific audience. There's this quote from Ginger Joe, who said that human beings have evolved into good intuitive statisticians and can gain insight, but only when information is presented simply and effectively. So what he's saying is that anybody can understand information if the information is presented in a simple and clear way. So we should really strive to do that and to present, however technical the results may be, into something which is very simple and clear that anyone can understand, because after all, you want this study to be read and understood. Another pitfall is that sometimes researchers tend to use results and move backward to define a research question. So we have this researcher who has a lot of data, a lot of information, and then he just starts analyzing it, getting some descriptive statistics, finding some associations between such and such, and then using that to drive a research question, using that to define a research question. The practice is known as data dredging or data fishing, p-hacking, like a fish expedition. Like we have this sea of data, and we just want to try to drag out the research question from that. And this is not proper. We need to make sure that we follow the normal pathway, leaving from the research question, leaving from defining a research question to actually answering the research question and reporting our results. Another pitfall is reporting only positive results, and this has led to this so-called publication bias, in which journals tend to report only studies that have statistically significant results. We need to remember that absence of statistical significance does not mean absence of clinical relevance. So reporting only positive results or the publication bias is really concerning, because it may lead to publication of deliberately falsified data by the authors. It may also lead to underreporting of relevant research. Another pitfall is finding spurious associations. Correlation does not mean causality. A correlation will just mean an association between an exposure A and an outcome or an event B. An individual who has been exposed to A is more at risk of developing an event B than another individual, but it does not mean that A causes B. To have causality, we need other factors, such as the strength of the association. For example, if it's a very high relative risk or a very high odds ratio, we need biological plausibility. We need to have a logical mechanism which could explain that A could possibly cause B. There's a constancy of the association. If this study was conducted in different population groups and in different time periods, will we obtain the same study results? There's also a temporal sequence. For example, like in cohort studies, it needs to be a study which has been done over a certain period of time, prospectively or retrospectively. So these are some criteria which define causality. So we need to ensure that we do not imply by our study results more than is actually supposed and allowed to imply. These are some tools which are useful for reporting results. You have the statistical analysis and methods in published literature. You have the International Committee of Medical Journal Editors, which provides recommendations for the conduct, report, editing, and publication of research. And we have the Equator Network, which contains guidelines for reporting various study designs. So, to conclude, what matters most is defining a good research question using the final criteria. The research question should be feasible, interesting, novel, ethical, relevant. Coming up with an adequate study design to answer the research question, and coming up with a rigorous methodology. If all these 3 steps are followed, then editors and reviewers will trust our results, whether the associations were found or not, and whether the associations were significant or not. So these are some tips which you may find helpful in reporting your results in a simpler and in a more engaging way and in a more compelling way. Thank you so much for listening.